Good morning, and thank you for joining. I would like to thank the organizers of the meeting for the opportunity to review these exciting posters on transcriptional reprogramming, ESR1 mutations, and novel targets and treatments in ER-positive breast cancer. These are the 4 posters that I will discuss, and I apologize in advance that I will not be able to show all the work presented in these posters given the time constraint. The estrogen receptor, or in short ER, is a key driver and therapeutic target in ER-positive breast cancer. The ER transcriptional axis is tightly regulated at multiple levels, and examples of some of the regulatory mechanisms that are tightly interconnected are ligand stimulation, post-translational modifications, crosstalk with receptor tyrosine kinases, and interactions with co-regulators. At the level of ER itself, genetic alterations can lead to aberrant ER transcriptional programs. The most common alterations are the activating missense mutations, with the Y537S and D538G being the most prevalent. These mutations lead to constitutive ligand-independent activity, and a number of years ago we showed that they have neomorphic properties leading to a transcriptional program that promotes pathways of inflammation and metastases and other pathways. In the clinic, the ER mutations are detected in at least 30% of patients with metastatic ER-positive breast cancer, mainly after treatment with an aromatase inhibitor. They confer resistance to aromatase inhibitors and relative resistance to fulvestrant. In addition, these mutations are prognostic of poor overall survival in metastatic ER-positive breast cancer. The crosstalk between ER and other nuclear receptors or other transcription factors regulates ER chromatin binding and, as such, regulates the ER transcriptional program. There are several modes of crosstalk that can lead to decreased ER activity, such as competitive DNA binding or the squelching of transcriptional machinery. This crosstalk can also increase ER activity with cooperative DNA binding, tethering, and chromatin opening by pioneer transcription factors, which facilitate ER binding. One of the key pioneer transcription factors in ER-positive breast cancer is FOXA1. FOXA1 opens condensed chromatin by displacing linker histone H1 and dictates a high proportion of ER binding. A number of FOXA1 genetic alterations associated with resistance to endocrine treatment were identified in clinical samples. These include FOXA1 mutations, mutations in the promoter region of FOXA1, and FOXA1 amplification. And this brings me to the first poster entitled Targeting the FRA1-Dependent Transcriptional Nexus in High FOXA1-Driven Endocrine-Resistant and Metastatic Breast Cancer, presented by Dr. Liu from Dr. Schiff's group. This group previously published work on a breast cancer cell model of acquired tamoxifen resistance in which FOXA1 amplification with FOXA1 overexpression was identified as a driver of tamoxifen resistance. They showed that FOXA1 overexpression leads to reprogramming of FOXA1 chromatin binding to superenhancer regions with an interplay between FOXA1 and the AP1 complex, and particularly the FRA1 and JUNB components of this complex, with or without ER binding. This in turn mediates the transcription of genes associated with endocrine resistance and metastases. In this study presented here, the group sought to investigate the AP1 FOXA1 centered transcriptional axis and its functional roles in endocrine resistant and metastatic breast cancer. First, by integrating RNA-seq and ChIP-seq data, they showed that FOSL1, encoded by FRA1, is the top gene from the AP1 complex that is induced by the reprogrammed FOXA1 binding in the tamoxifen-resistant cells. In a second integrative analysis, they identified FRA1 itself and the embryonic transcription factors SOX9 and KLF4 among the key transcription factors induced by the gain FOXA1 binding sites and regulated by FRA1 and JUN. As shown in these Western blots, they confirmed at the protein level that these genes are indeed upregulated in the tamoxifen-resistant cells and that SOX9 and KLF4 were downregulated with the silencing of FRA1. They next looked at the overlap between the human secretome and the genes regulated by FOX1, FRA1, and JUN and identified a secretome of 25 genes. and when looking at RNA-seq data from clinical samples, they found that the secretome 25 is upregulated in metastatic versus primary ER-positive breast cancer tumors. Lastly, silencing of FRA1 reduced key protumorigenic and metastatic phenotypes, including clonogenicity, soft agar colony formation, and migration. To conclude, these results indicate that FRA1 is the top AP1 component upregulated by high FOXA1 engaged superenhancers in tamoxifen-resistant cells, and FRA1 potentially forms a feed-forward transcriptional axis to amplify high FOXA1-induced enhancer reprogramming that activates a pro-metastatic secretome. As such, the authors propose that FRA1 is a potential therapeutic target. And these are a few questions for discussion. Is this model seen in resistance to other endocrine treatments? Is the activity of FRA1 dependent on FOXA1? And how can we target FRA1 for therapeutic purposes? The next poster focuses on the androgen receptor, or in short, AR, which is expressed in about 90% of ER-positive primary breast cancers, highlighting the importance of this nuclear receptor in ER-positive breast cancer. A recent study published earlier this year showed that AR activation in the presence of estradiol leads to the reprogramming of ER binding, resulting in the repression of ER-regulated pro-proliferated genes through the squelching of co-activators and ultimately resulting in tumor suppression. In line with this study, a second study showed that AR is an ER inhibitor, and AR expression is associated with good outcomes in ER-positive breast cancer. Importantly, promising results were seen in a phase 2 study of an AR agonist, enobosarm, in ER-positive metastatic breast cancer. And based on these results, there is an ongoing phase 3 study of enobosarm. However, previous studies showed that AR inhibition reduced estradiol-mediated proliferation, and AR overexpression is associated with endocrine resistance. These conflicting results may be due to context-dependent activity of ER, and this poster presented by Dr. Grimm from Dr. Fuqua's group entitled Mutant ESR1 Receptors Antagonize the Tumor Suppressor Function of Androgen Receptors. The authors aim to define whether AR has a role in proliferation or metastases, or tumor suppression in the context of the ER mutations. To start, they looked at the transcriptomic analysis of ER mutant and wild-type cells in hormone-deprived, estrogen-stimulated, or androgen-stimulated conditions. As expected, gene set enrichment analysis showed that the mutant cells had increased estrogen signaling in hormone-deprived conditions. Interestingly, in estrogen-stimulated conditions, the androgen response was reduced in the ER mutant cells compared to wild-type cells. The authors went on to do ER and AR chip sequencing in wild-type and ER mutant cells in hormone-deprived and estrogen-stimulated conditions, and showed that in the presence of an ER mutation and in estrogen conditions, AR binding was redistributed when looking at all AR peaks and also the sites that have ER and AR co-binding. Additionally, motif analysis showed that ERE was the only motif enriched in the ER-AR shared peaks. When looking at the overlap between the genes associated with the ER mutant ER-AR co-binding peaks and the genes differentially expressed in the mutant cells, they identified a gene set of 65 genes, and these genes were associated with worse outcomes in primary ER-positive breast cancers. Furthermore, several of these genes had increased expression in metastatic versus primary breast cancers. Lastly, when applying published ER chip sequencing data of ER-positive breast cancer tumor samples, the authors showed that the ER binding density of the mutant ER-AR shared binding sites in E2-stimulated conditions was highest in tumor samples of metastases. The authors conclude that these results suggest a two-pronged genomic activation mechanism in ER mutant tumors with AR redistribution and genomic co-localization of AR and ER binding sites. They propose that loss of AR tumor suppressor function may help metastases in ER mutant tumors through collaboration between AR and mutant ER. A few questions that come up. How do the ER mutations affect ER in the presence of AR agonists? and the ER mutation phenotypes are usually observed in E2-deprived conditions. How do the authors explain that the AR redistribution was seen in E2-stimulated conditions? And lastly, will the presence of an ER mutation impact clinical benefit from enobosarm? The next poster presented by Dr. Lee from Dr. Oistreich's group is entitled ESR1 Mutant Breast Cancers Show Elevated Basal Cytokeratins and Immune Activation. In this poster, the authors asked if the ESR1 mutations that promote metastases confer a molecular subclassification shift and induce immune activation. To answer this question, they looked at the expression of the PAM50 genes in wild-type and mutant ER cells and 97 breast cancer cell lines. As shown in this PCA plot, the mutant cells clustered with the basal cell lines. The mutant ER cells had higher expression of basal signatures and lower expression of luminal signatures when testing other signatures of the intrinsic molecular subtypes. In addition, in metastatic breast cancer tumors harboring the ER mutations compared to wild-type ER tumors, there was higher expression of basal signatures. When looking at the basal genes upregulated in the mutant cells and mutant tumors, they found that basal cytokeratins were the leading enriched basal markers and validated the upregulation of these genes in ER mutant cells by quantitative RT-PCR. They next showed that the expression of these genes is not dependent on ER. As shown here in these genomic tracks of ER chip sequencing in a locus on chromosome 12 that includes a number of the basal cytokeratins, there was no ER peaks in wild-type or mutant cells. In addition, silencing of ER led to actually an increase of basal cytokeratins in both wild-type and mutant cells. Interestingly, immunofluorescence staining showed that the basal cytokeratins were expressed in the cells with low ER expression. Next, using publicly available epigenetic data of MCF7 cells, they showed that a number of the basal cytokeratins are located within a genomic locus on chromosome 17 that has CTCF and cohesin binding, as well as CTCF looping and a superenhancer with PR binding. They validated enrichment of CTCF binding in the presence of the ER mutations by CHIP PCR. Additionally, they showed that the basal cytokeratins is dependent on progesterone receptor since their expression was decreased with silencing of the progesterone receptor. Lastly, the authors show that immune-related pathways are commonly enriched in the ER mutant tumors and other ER-positive tumors that have high basal cytokeratin expression. S100A8 and S100A9, which heterodimer and form a key pro-immune cytokine, were the leading immune-related genes enriched in these tumors. And indeed, the concentrations of the S100A8-9 complex was higher in blood samples from patients with metastatic breast cancer who were found to have an ESR1 mutation by cfDNA analysis. Taken together, the authors propose a working model in which, in the presence of the ER mutation in cells with low ER expression, PR activation of a superenhancer upregulates the transcription of basal cytokeratins through a CTCF-driven chromatin loop, and this is associated with increased expression of key cytokines. Some questions for the authors. Do the cells that have low expression harbor and express the ER mutation? And could there be a paracrine effect between cells harboring the ER mutation and wild-type ER cells? What is causing the activation of the PR-associated enhancers? Is this levels of PR, crosstalk with other transcription factors, post-translational modifications, or other co-regulators? And lastly, is the CTCF looping different in mutant versus wild-type cells? Moving to the last poster entitled Constitutively Active Estrogen Receptor Mutants Enhance Breast Cancer Pathogenesis by Co-opting Progesterone Receptor Activity, which can be countered by giredestrant, presented by Dr. Liang from Dr. Metcalfe's group. Since this poster continues the theme of the role of the progesterone receptor in the presence of ER mutation, let's briefly review some other aspects of the PR-ER crosstalk. We all know that PR is an ER transcriptional target. It is less known that PR interacts and reprograms ER binding, and downstream consequences are likely context-dependent, since on one hand, PR activity has been shown to be associated with good clinical outcomes, and copy number loss of PR is associated with worse outcomes in ER-positive breast cancer. Additionally, PR agonists have been used for the treatment of metastatic ER-positive breast cancer. In contrast, other data show that PR antagonists decreases proliferation in ER-positive breast cancer. In addition, in the Million Women study, the risk of developing breast cancer was higher in women who took hormone replacement therapy with estrogen and progesterone versus estrogen alone. However, it is important to note that the increased risk of breast cancer likely depends on the specific type of progestin. Now, given the significance of the ER mutations, an important question is, what is the role of PR in the presence of these mutations? As a reminder, in the previous poster, we saw that PR increased basal cytokeratins in mutant ER. And now let's go back to the last poster. The aim of this study was twofold. First, to develop an in vivo model system to investigate the in vivo biological properties of the ER mutations. Second, to establish the in vivo sensitivity of mutant ER phenotypes to giredestrant, a latest generation ER antagonist and degrader. To this end, the authors generated conditional knock-in models with the mouse equivalent of the human D538G and Y537S mutations. Constitutive ER activity was evidenced by the high correlation between genes upregulated in the mutant model in ovexed mice and E2 upregulated genes in wild-type mice, both compared to wild-type ovexed mice. Examination of the mammary glands revealed that the ER mutations drive increased cell proliferation, budding, and branching. However, these phenotypes were seen in only about 50% of the mice, and upon further investigation, the authors discovered that these phenotypes were only seen in the diestrous phase of the estrous cycle, which is the phase that is high in progesterone. Furthermore, in ovexed mice, these phenotypes were not induced by estrogen alone, but were induced by progesterone with or without estrogen. CHIP sequencing studies showed that the ER mutations led to the reprogramming of ER and PR binding in the mammary glands, and the ER mutants alter response to progesterone as evidenced by a subset of progesterone-responsive genes with increased expression in the mutant models. Additionally, this effect was mutant-specific. When ovexed mice were implanted with estrogen and progesterone, a PR antagonist, or the ER antagonist giredestrant, prevented the ER mutant phenotypes. In addition, in 2 PDX models harboring the ESR1 mutations, progesterone, as shown in the red line, stimulated tumor growth, and this was reversed by either ER or PR antagonism. In conclusion, the ER mutations alter the transcriptional landscape and proliferative capacity of mammary cells beyond what can be attributed to estrogen-independent activation of wild-type ER program, thus neomorphic properties that arguably have tumorigenic phenotypes. The ER mutations can alter responsiveness to other endogenous hormones, and in this case, progesterone. These ER mutant phenotypes are ER-dependent as they are abrogated by ER antagonism, and tumor growth in PDX models harboring the ER mutations is also enhanced in response to progesterone and decreased in response to ER inhibition. A few questions to the authors are, what is the overlap between PR and ER gain binding sites, and what are the motifs in these gain sites, and which transcriptional signatures are enriched? What is the effect of the ER mutations on the immune and other stromal cells? And importantly, do these mice develop tumors? And with this, I will end and wanted to thank all the authors for their excellent posters and also thank you for your attention.